This tutorial will walk through this problem about population genetics. Please pause the video for a minute to read through the problem, then come back when you're finished to see how we're going to approach it. So the first question here asks if the population is in Hardy-Weinberg equilibrium. Before we answer that question, let's review what it means for a population to be in Hardy-Weinberg equilibrium, sometimes referred to as HWE. HWE is a theoretical model that describes populations that are not evolving. In other words, populations can be in HWE, in which case they aren't evolving, or they can be out of HWE, in which case they could be evolving. Population genetic analyses can look gene by gene to see if, for a given population, changes in the frequencies of different versions of a gene are occurring. Changes in these frequencies are a driving force of evolution because they lead to changes in the physical characteristics exhibited by members of the population. So let's start with the theoretical model. What happens if a population is in HWE? Well, this means that the population fits certain characteristics, and as a result, allele frequencies will not change over time, genotype frequencies will not change over time, and genotype frequencies can be predicted accurately based on allele frequencies. For example, if, for a particular gene, two alleles, big A and little a, are possible, and in a population, the frequency of big A is said to be represented by P, and the frequency of little a is said to be represented by Q, then the frequency of the big A homozygote is P squared, the frequency of the little a homozygote is Q squared, and the frequency of the big A heterozygote is 2 times P times Q. So what are the characteristics of a population for which these predictions will hold? There are 5 so-called requirements for a population to be in HWE. There can be no mutation, migration, or selection, and this no selection includes both selection that affects survival and selection that affects reproduction, like mate choice. In addition, mating must be random, and the population must be large, theoretically infinite. Practically speaking, populations start to act as though they are infinitely large, at least in the short term, once they get up to about 10,000 members. So now, let's go back to our question. Based on the data in the table, is the population in Hardy-Weinberg equilibrium? The answer here is no. If a population is in HWE, neither the genotype nor the allele frequencies will change over time. In this population, we can see just by eye that the genotype frequencies are changing. The frequencies of big F, and little f homozygotes have increased in the 20 generations we're analyzing, and the frequency of big F, little f heterozygotes has gone down. We don't know if allele frequencies are changing yet, but genotype frequencies certainly did, and that's all we need to know that HWE is not in effect. Okay, so on to the next part of the question. Calculate the genotype and allele frequencies in generations 0 and 20. Let's start with generation 0. The frequency of the big F allele is equal to the total number of big F alleles divided by the total number of alleles. In other words, the number of big F alleles in big F homozygotes plus the number of big F alleles in heterozygotes divided by the total number of alleles, which is twice the total number of individuals. So we have 2 times 26,000 big F alleles in homozygotes, 28,000 big F alleles in heterozygotes, and 200,000 total alleles. 80,000 divided by 200,000, or a frequency of 0.4 for the big F allele. For the little f allele, you can either subtract the big F frequency from 1 or do the calculation again. I like to repeat the calculation as a kind of double check. So the total number of little f alleles will be 2 times the number of homozygotes, 46,000, plus the number of heterozygotes, 28,000. You divide that total by 200,000, the total number of alleles, to obtain a value of 0.6 for the frequency of the little f allele. And in generation 20, we can do the same thing. The frequency of the big F allele is 2 times 30,000 homozygotes plus 20,000 heterozygotes divided by 200,000 total alleles, or 0.4. The frequency of the little f allele is 2 times 50,000 homozygotes plus 20,000 heterozygotes divided by 200,000 total alleles, 0.6. So what we can see here is that even though the genotype frequencies were changing, the allele frequencies were not. This doesn't mean that the population is in HWE. It isn't, since genotype frequencies weren't stable, as we saw before. So now, we can start thinking about what could explain this. In order to figure out what characteristic of the population could be causing this non-equilibrium behavior, It's useful to compare our observed genotype frequencies with the ones we could expect if HWE is in effect. So that's the next part of the question. Based on the allele frequencies that you calculated, what are the expected genotype frequencies in each generation? For this, we'll go back to our genotype frequency formulas, but since the allele frequencies for both generations are the same, we only need to do the calculation one time. Recall that the frequency of the big F allele is 0.4, and the frequency of the little f allele is 0.6. We'll designate the frequency of big F as P and little f as Q. You can switch the P's and Q's if you want, as long as you clearly define which one's which. Okay, so the predicted frequency of the big F homozygote is P squared, or 0.4 squared, which is 0.16. The predicted frequency of the little f homozygote is Q squared, or 0.6 squared, which is 0.36. And the predicted frequency of the heterozygote is 2pq. So that's 2 times 0.4 times 0.6, or 0.48. Easy peasy. Now comes the part that's a little more challenging. The next question asks, deviation from what two Hardy-Weinberg assumptions could best explain the discrepancy between the observed and expected values for this population? For this, now we need to see how our observed values differ from what we expect. If we have a sample of 100,000 individuals, if our expected frequency of big F homozygotes is 0.16, that's 16,000 individuals. If our expected frequency of little f homozygotes is 0.36, that's 36,000 individuals. And if our expected heterozygote frequency is 0.48, that's 48,000 individuals. So when we compare the numbers, we can see that we've got more homozygotes of both types than expected and fewer heterozygotes. This discrepancy is greater in generation 20 than it was in generation 0, but in both cases, we do see a similar result. So what could cause this? Let's look at each deviation and see which provide possible explanations. First, no mutation. Well, we can cross this one off right away. We know that mutation is very rare, and so wouldn't be able to explain a change this significant in 20 generations. No migration? We're told that this population is in an isolated lake, so we can cross this one off too. No selection. For this, we want to think about the physical characteristics of the organisms themselves, and if the changes that we're seeing are consistent with one or more of them being advantageous or deleterious. Here, what we see is more than expected big F individuals, more than expected little f individuals, and fewer than expected big F individuals. Since all three genotypes correspond to distinct phenotypes, it looks like being of an intermediate size from the big F genotype is selectively disadvantageous, since we see fewer of these than we expect. Correspondingly, being either large or small is advantageous. So selection is one possibility that could explain the data. Next, we have random mating. Let's compare that with what we see. More homozygotes than expected, fewer heterozygotes, and no change in allele frequency over time. This is a classic characteristic of inbreeding and other types of non-random mating, where individuals are more likely to mate with others like themselves. So if, for example, small fish are more likely to mate with each other, and not likely to mate with large fish, all those small f alleles from the small fish parents combine with each other to make more small f offspring. You end up losing out on the heterozygotes, because if small f fish don't mate with large f fish as much as you'd expect based on random mating, you get fewer little f gametes combining with big F gametes to make heterozygous offspring. So now we have two possibilities that fit the data, but let's just talk about the last characteristic to make sure it doesn't apply: large or infinite population size. Now, no population is actually infinite in size, but this one is pretty big. Our sample is of 100,000 fish. If populations behave as though they are infinite at 10,000, then this is certainly large enough to qualify, so we can cross this possibility off our list. And here we have it, two Hardy-Weinberg assumptions that could be violated in this population to produce the deviations that we see. And now, for the last part of the question. A new residential property is being built by this lake, and the developers have created a number of small ponds around the property. They catch several batches of fish from the lake to seed the ponds. What HW assumption is likely to be violated in the ponds? And what is the name of this violation? The ponds are described as being of small size and will violate the large population assumption. In particular, since the developers are catching different batches of fish from the lake to seed the ponds, they are generating a different founder population for each small lake. Another way of putting this is to say that you'll see the founder effect in the small ponds. This is one type of population bottleneck, which is another way that you could describe this scenario. So here we have it. We don't know for sure what's causing the change in genotype frequency in the lake, or what will necessarily happen in the ponds, but we can say that there's something going on there, and it might be worth studying more to figure out exactly what it is.